So next topic is vector. Now we'll quickly revise the steps in recombinant DNA technology and why is vector needed, why is vector so much important in recombinant DNA technology. So recombinant DNA technology is combining two different DNA molecules or genes of two different organisms together, forming a new DNA which is not naturally present, known as recombinant DNA technology. This new DNA, recombinant DNA formed, is unique. These are the applications of recombinant DNA technology. Is used for betterment of crops, for recombinant vaccine production, for prevention and cure of different diseases like cystic fibrosis, sickle cell anemia, for production of industrially important proteins that can be used as pharmaceuticals like monoclonal antibodies, clotting factor, blood clotting factors, insulin, and so on. So we'll move ahead with the steps in recombinant DNA technology. First is isolation of gene using restriction enzyme. Now this isolation of gene means the gene of interest. Say if you want to produce insulin at larger amount, if you are interested in large scale production of insulin, if you are interested in large scale production of antibodies, that particular gene you have to isolate by cutting it with a restriction enzyme from particular organism's DNA. Okay, so remember, before this isolation you have to isolate DNA of an organism, then you have to cut the gene of interest out of it, then you have to attach this gene of interest or say insert to the vector. Vector is the gene carrying vehicle which will carry this particular gene of interest to the particular host organism. Okay, so the transfer of this gene to the organism in which its production is desired is carried out by a vector. So the vector acts as a vehicle that transfer this gene to the host organism. So the host cell now will multiply this gene of interest, will replicate this gene of interest with the vector molecule and then produce a product from this. Product is RNA first and then the proteins are formed. This produces many copies of host cell and host cell consists many copies of particular product. And then last step is extraction or separation or purification of product of gene of interest. So this is insulin gene which is cut open with the restriction enzyme. This is isolated using restriction enzyme like this. This insulin gene is separated. Then this vector, bacterial plasmid, is used as a vector here. Now there are different types of vectors that are artificially formed in laboratories by joining different segments of the DNA together, which forms artificial vector. So bacterial plasmids are used as vector by removing certain sequences from it and joining this gene of interest instead. Okay, the sequences which are not required, they are removed from the bacterial plasmid and this gene of insert is attached to it. Now since this is circular DNA molecule, when you cut open it with the restriction enzyme, that will generate linear DNA molecule. Okay, and you have to attach gene of interest to it. Okay, then this vector carrying gene of interest. Now gene of interest here is insulin, is transferred to the host organism. The process is known as transformation. transferring of gene of interest in organism in which it is not naturally present but will support its multiplication and production of protein from it. Okay, no, such organism is known as host organism. Then host is allowed to multiply along with the host chromosomal DNA as well as this plasmid DNA will multiply, and your gene of interest is expressed in the form of protein, that is insulin is produced. Then these host cell, after their growth, they are lysed and insulin protein is extracted or purified from it. These are basic steps in recombinant DNA technology. Now we are going to discuss different types of vectors that are used in this process of transformation or carrying gene of interest to the host organism for its large scale production. Okay. So these are different DNA cloning component. One is gene of interest, then restriction enzyme, vector, DNA ligases, antibiotic, and a host organism. The vectors. Vector is an agent that carry a gene fragment into a host cell, a gene into a host cell. So this acts as a vehicle. The vector is defined as gene carrying vehicle. The vector are the DNA molecule that can carry a foreign DNA and replicate inside the host cell. So the vectors are of different types. One is cloning vector and another one is expression vector. Cloning vectors are used only for getting multiple copies of gene of interest. Okay, cloning vector, it is used only for generating number of copies of gene of interest for say preserving those genes or for preparing DNA libraries. And expression vectors are used for expressing particular gene of interest. Okay, so there are different types of vectors which can be used like plasmid, bacteriophages, cosmid, bacterial artificial chromosomes or BAC, yeast artificial chromosome, YAC, retroviruses, baculoviruses, agrobacterium plasmids, they can also be used as vectors. So vector choice depends on the choice of host organism. Okay, that host should support the replication of vector molecule. So this vector should have following properties. It must be easy to isolate and smaller in size. It should be non-toxic to the host cell. That is, it should not produce any protein that can hamper, that can reduce the growth rate of cell or that can stop metabolism of host cell. It should not be toxic. Then it should have a space for foreign gene insert. Remember, smaller the size of vector, larger the gene of interest you can attach. जितना छोटा वेक्टर होगा उतना बड़ा gene of insert you can attach. Okay, and that vector should not have any gene encoding antibiotic that can kill host cell, or that should not encode any proteolytic enzyme that kill host cell. It should not be toxic to the host cell. Then it have space for foreign insert. Then it must be capable of autonomous replication. Autonomous replication, that is, it should replicate independently of the main bacterial chromosome. That is, when it is transferred into the host cell, its replication, vector का replication must not be dependent on host cell's DNA replication. Vector DNA should replicate independently so as to have multiple copies of vectors produced per cell. एक सेल में मल्टीपल कॉपीज प्रोड्यूस होनी चाहिए, इसलिए ऑटोनॉमसली रेप्लिकेटिंग वेक्टर होना चाहिए. ओके. हैव वेक्टर शुड हैव यूनिक रेस्ट्रिक्शन साइट्स for common restriction enzyme. So one vector, if you consider, that should have multiple restriction sites or multiple cloning site. That means for one restriction enzyme it should have one site, and such many sites must be present on vector molecule for different enzyme. एक restriction enzyme के लिए only one site, but multiple sites for different restriction enzymes. It should have convenient marker for selection of transformer. For example, antibiotic resistance gene. Now this vector must have the selectable marker gene. What do you mean by selectable marker? Is when this vector is transferred to the host organism, host cells must be identified which have these vector molecules present in it. Okay, that is known as selectable marker gene. These genes allow host organism to be identified which have the vector molecule in them. Okay, जिनमें ट्रांसफर हुआ है, such cells are known as transformant, and these transformants, how to identify such transformant depends on the selectable marker gene that we'll discuss later on. Okay, vector must be relaxed, that is it should produce multiple copies in a host cell. Okay, multiple copies in host cell. So the very first vector is plasmid vector. Plasmids vectors. Now what are plasmids? Plasmids are covalently closed circular double-stranded DNA that are present naturally and replicate autonomously in bacterial cells. Okay, plasmids, they have their own origin of replication and they have a drug resistance marker. Vectors, plasmid vectors are prepared from natural plasmids by removing unnecessary segments and adding essential sequences. Bacterial vectors are extracted, they are cut open to remove unnecessary segments and add extra sequences required for transformation and production of a protein from gene of interest are added or attached to the plasmids, and then the artificial vector is formed. Typically, a plasmid vector has a polylinker gene which can recognize several restriction enzymes and ampicillin resistance gene for amplification. Now this ampicillin resistance gene is selectable marker and a origin of replication for its autonomous or independent replication. Now see here, this plasmid vector, it has origin of replication which helps in replication of this plasmid. Then this has a marker gene, resistance gene. Then see here, you can see multiple cloning sequences. These are known as multiple cloning site, MCS. That is, here are many restriction enzyme sites. Any one you can use for attaching your gene of interest. And promoter is required for transcription. Plasmid vectors are modified to contain a specific antibiotic resistance gene and multiple cloning site, also called as polylinker region, which has number of unique target sites for restriction endonucleases. For uh, for many enzymes, only one site is present on this vector. Cutting the circular plasmid vector with one of these result in single cut, creating a linear fragment of plasmid. Okay, a foreign DNA molecule referred as insert cut with the same enzyme which you are using for cutting vector molecule can be joined to the vector in ligation reaction with the help of ligases enzyme. The bacterial plasmids cannot accept DNA larger than 5000 bases or 5 kilobase pair. Hence, they are restricted to cloning DNA less than 5000 base pairs. Now see, here this plasmid vector consists site of for EcoR1, and this green colored one is the gene for antibiotic resistance, or this is selectable marker gene which will be used further for isolating transformants. Okay, this is foreign DNA. This is gene of interest shown with the pink color. So here, for cutting open both DNAs, vector DNA and this foreign DNA, same enzyme is used, EcoR1. Which sequence is recognized by EcoR1? It is GAATTC. We have to remember, at least remember 5 restriction enzymes names with their recognition sequence and cut site or cleavage site. Okay, then once the plasmid DNA and this gene of interest are cut, they have similar ends, sticky ends, because they have been cut with the same restriction enzymes. So they are ligated together and this recombinant DNA is formed. Okay, then this recombinant DNA is inserted in an host organism, into host organism. Now inside this host, this vector recombinant DNA replicates like plasmid and generate multiple copies of it per cell. Okay, now since here this is antibiotic resistance gene, now only few cells will get this foreign DNA, recombinant DNA catch up only. See, remember, this recombinant DNA when transferred to the host cell, only few host cells will catch up this DNA. Other cells won't accept this DNA. Not all the host cells accept this foreign DNA or recombinant DNA inside them. The one which accept them, they are separated or isolated using this antibiotic resistance gene. Say for example, here is ampicillin resistance gene present on this plasmid. The host initially is ampicillin sensitive. That is, if you add ampicillin into the medium, the host cell won't grow on it because host is ampicillin sensitive. It do not have any mechanism to protect itself from ampicillin antibiotic. So such host cells are killed. Now these host cells which are transformed, these have this ampicillin resistance gene present in them because they have this recombinant DNA. So host cells after transformation, they are grown on the medium containing ampicillin. जो भी मार्कर जीन होगा वो ऐड करके you can select recombinants, eliminating non-recombinants or non-transformed cells, because now this ampicillin resistance gene produce a protein which degrade ampicillin, and therefore these host cells are not hampered by the presence of antibiotic ampicillin. Okay, so only bacteria containing recombinant cell DNA will grow on this antibiotic containing gene. See here you can see these two cells are shown with pink color because they have foreign gene or vector DNA in them. They are transformed, and these white one, they are not transformed. They won't grow on this ampicillin containing medium because they are antibiotic sensitive. These cells which have acquired this resistance for ampicillin is because of presence of this foreign gene. Okay, so this is a selection method using this marker gene. So that's why this type of marker is known as selectable marker, because this marker gene selects only transformants or allow only transformants to grow. Okay, then after their growth the DNA is purified, and then again the copies of DNA are extracted. If you are interested in protein production, you can extract, purify proteins out of it. Okay, so the same picture is given here. Same is given here with the help of replica plating method. Then the first plasmid vector is PUC19. PUC19. See, this P you have to write with a small letter. P stands for plasmid. UC stands for University of California, because in University of California scientists developed this vector, and 19 is its identification number. Okay, so this PUC19 is a cloning vector. P stands for plasmid. UC stands for university in which it was created. It is artificially created vector. So it is circular double-stranded DNA which has 2686 base pairs. See, you have to remember these base pairs for each vector, and the size of insert it can accommodate. You have to remember all these things. Size of the vector, size of the insert it can accommodate, and different genes present on it. So this PUC19 has ampicillin resistance gene, LAC-Z alpha gene, and origin of replication. LAC-Z alpha gene encodes N-terminal fragment of beta galactosidase. Okay, beta galactosidase. This beta galactosidase, if you remember lacoperon. Okay, lacoperon encodes beta galactosidase for utilization of lactose sugar in absence of glucose. The multiple cloning site region. See, See, this is multiple cloning site region. It's split into lag Z gene where various restriction sites for many restriction endonucleases are present. Here you can see different restriction enzymes. You can use any one of it to cut it open. The origin of replication is derived from PMB1 vector. It is small vector but has high copy number. The lag Z gene encode beta galactosidase enzyme. So this is multiple cloning sequence. So whenever the question is asked in examination, draw a map of plasmid vector, any one plasmid vector. You have to draw a circle like this and show such different genes with the number of size of this vector. Okay. Now this plasmid PUC19, this have lag Z gene used for blue-white screening. Lag Z gene is derived from lag operon. Okay. When given an activator IPTG, IPTG stands for isopropyl thiogalactoside, and XGAL, XGAL stands for, this is long form of XGAL, 5-bromo-4-chloro 3-indolyl beta D galactoside, gives blue colonies. When the lag Z gene is present and you provide IPTG and XGAL into the medium, if added, these two, this activator activates this lag Z gene. Once the lag Z gene is activated, it produces a protein or enzyme beta galactosidase, and this beta galactosidase cut this XGAL substrate. This XGAL is a substrate for beta galactosidase which develops blue color. So the colonies which develop blue color, they are transformants because they have this lag Z gene, and lag Z gene is present on PUC19 vector, to which you have attached your gene of insert, gene of interest. Okay, so the blue colored colonies are transformed and white colored are non-transformed. These activator activates operon, so lag Z gene is transcribed and lag Z protein is produced. This combines with the chromosomal DNA transcribed protein and forms hybrid beta galactosidase. This hydrolyze XGAL, which is chromogenic substrate, and releases blue color. When no active lag Z gene, when lag Z gene is not active, you will have white colonies. The non-transformed host, they show white colonies, and the transformed, they show blue colored colonies. And this procedure is known as, this method for selection of transformants is known as blue-white screening or alpha complementation method. Why alpha complementation? Because this PUC19 has only lag Z alpha gene which encodes N-terminal of beta galactosidase, and rest of the part of beta galactosidase is derived from chromosomal DNA of the host cell. Okay, so it is known as alpha complementation. It is complementing the defect of host cell, complementing the host cell by providing N-terminal of beta galactosidase or blue-white screening, because blue and white able you to differentiate between transformants and non-transformants. Then the PUC19 second vector, plasmid vector, PUC 18. So both are same, only the difference is, see this PUC 18 is small, 2.7 kb, almost similar in size, can accommodate 5 to 10 kb insert. It has high copy number per cell. One cell may have 500 PUC copies. Multiple cloning sites are there. It has multiple cloning sequences for 13 different enzymes and ampicillin resistance gene. Now only the difference in PUC 19 and 18 is in selectable marker. Here LAGZ alpha gene is present as selectable marker on PUC 19, and on PUC 18 ampicillin resistance gene is present along with the LAGZ alpha. LAGZ, this is complement sign. LAGZ complement. This is the map of PUC 18. This has origin of replication, LAC-I gene which is inducer, LAGZ alpha, then multiple cloning sites and ampicillin resistance gene. Then PBR322, third plasmid vector, PBR322. This is larger in size than PUC 18 and 19. This has size is 4300 base pair. This has two selectable markers. One is ampicillin resistance and another one is tetracycline resistance genes. 20 unique restriction enzyme sites are present on it. Okay, 20 restriction sites are present. Two selectable markers. Origin of replication. BR. Now this PBR322 name stands for plasmid, developed by Bolivar and Rodriguez. BR, these are names of scientists who created this plasmid, and 322 is its identification number of this vector in their lab. Size is 4362 base pair. Copy number is 15 to 20. Two recognition sites are present within tetracycline resistance gene. See this BAMH1 and SAL1. Within the tetracycline resistance gene, there are two restriction sites present, and within ampicillin resistance gene, there are three sites present. Use any one for insertion and other for selection. That is, if you are using this PST1 for cutting open this plasmid, this ampicillin, and attach your gene of interest here, so this ampicillin resistance gene will not be functional. Okay, it will lose its function because you are attaching gene of interest here in between. So no protein is formed which will prevent the cell against this antibiotic, ampicillin antibiotic. So this if it is transferred into the host cell with the gene of insert attached here in ampicillin resistance gene, you have to use selectable marker, this one, tetracycline resistance gene. If you are attaching your gene of interest here within tetracycline resistance gene, then you have to use ampicillin resistance gene as selectable marker, because if you attach gene here in tetracycline resistance gene, this tetracycline resistance gene will not be functional because its sequences are changed. So no protein is formed which will prevent the cell against tetracycline antibiotic. But you can use ampicillin resistance gene for selection of transformants. Okay, this one will discuss in next lecture, agrobacterium.